Hello, I would like to thank the organizers for organizing this workshop, which is very mechanistic in nature, very interesting topics. And I'm happy to share our most recent data on how RNA Polymerase II communicates with the nucleosome, how it goes through a nucleosome, but also have some surprises at the end, some new topics that are coming up. Before I get started, I would like to ask you that if you're interested, that you join the talk by Goran Kokic on Friday, because he will present our latest work on the coronavirus polymerase structure, as well as a mechanism for how the drug remdesivir can stall the polymerase. So about 10 years ago, our great colleague John Liss has summarized the key steps in the beginning of RNA polymerase II transcription, which are subject to regulation in eukaryotic cells. So this starts with chromatin opening, then a pre-initiation complex forms, the DNA opens, and often in metazoan cells, the polymerase would pause just downstream of the transcription start site, and then factors are exchanged until the polymerase is released into active elongation. And then it also carries factors with it to go through nucleosomes, which is a very exciting mechanism, because you have to imagine that polymerase is at the speed of about 3,000 or so base pairs per minute, would actually go through, let's say, 15 or so nucleosomes per minute. So today I will concentrate on this last step, but I would like to tell you that we have just written and published a new review where our work from the last 2 decades is reviewed, but of course also the work from many other laboratories who have worked on this problem of RNA polymerase II transcription mechanisms. We have tried to serve the community by solving 3-dimensional detailed structures that elucidate the mechanisms that occur at each of these steps at the beginning of the transcription cycle. Now, how does transcription begin? As you all know, we start with formation of a large pre-initiation complex, and then the polymerase escapes from the promoter. And after about 50 or so base pairs, it will often pause, at least in metazoan cells. And that pause, we know from the work of many labs, is stabilized by the negative elongation factor. And that paused elongation complex needs to be converted into an activated released elongation complex, which requires the PTEF-B kinase complex that contains the CDK9 kinase. And then you end up with this activated complex. And we found that in vitro, at least, you need both SPT6 and the PAF complex to make this more efficient. Now that we had a biochemical system for investigating these steps, we could also reconstitute all the complexes. And over the years, we were able to determine the 3-dimensional structures through a combination of X-ray crystallography and, over the last years, of course, cryo-electron microscopy. And now we have a picture of the pre-initiation complex, which is actually the yeast species, but also of the pause elongation complex and the activated and released elongation complex that we call EC star. And these two complexes are made from mammalian and human proteins. Now, we found through a combination of structural biology and functional assays how the paused complex is actually adopting this pause state. Just briefly, it will bind to a tilted DNA-RNA hybrid, and the NELF factor is stabilizing this conformation of the polymerase and reducing mobility of the polymerase so that this paused complex is more longer lived. We have also used functional genomics in vivo in human cells to investigate this switch at the beginning of genes And briefly what we found is that we could recapitulate what other labs had reported, namely that the PTEF-B kinase complex, CDK9 in particular, is important to reduce the duration of polymerase pausing and rapidly convert the paused complex to an activated complex. But very interestingly, we additionally found that if you change the duration of pausing, you will actually influence the rate of productive initiation. So the step of pausing is setting a limit to the rate of productive initiation, which is very interesting because it explains how at the step of pausing you can regulate the output of genes, namely the number of mRNA molecules made per time, which is of course dependent on the number of initiation events and successful processive elongation to the end of the gene. Now, when you plot MNase data at the pause site, you will find that just downstream of the site of pausing, there's a position +1 nucleosome. And because we have these distances from these in vivo data, we can come up with a model of the situation at the promoter and just in the promoter proximal region. This is a model, but it basically shows you that you can accommodate the pre-initiation complex, the paused complex, and then the downstream nucleosome. And you can appreciate that on the one hand, it may be sterically possible that the paused complex is inhibiting initiation or DNA opening because clashes will form. And that was originally suggested by Jesper Svejstup's lab. And on the other hand, you can appreciate that you do need to exchange factors at this point because you need to go through the +1 nucleosome. So you do need to load factors for chromatin transcription. Now, we also, about 2.5 years ago, maybe 3 years ago, we solved the structure of the polymerase and complex with the nucleosome just downstream of the polymerase. And please compare the great work by Sekine and Kuramisaka from Japan. They have in the same year published structures, similar structures in a different yeast species. And I'm sure we will hear about that later on. Now, this was a good basis to look at the way that transcription factors can promote transcription elongation through nucleosomes. And let me start with this model here, where we just superimpose our activated complex EC star with our polymerase nucleosome complex to show that the nucleosome is actually accommodated, or in other words, the elongation factors are accommodated in the presence of the nucleosome. And the question is, of course, now, what is the role of these factors? So we have begun to investigate this in more detail. Of course, there's some literature, but we now can really go into the details. And we looked at SPT6 in human cells, and we asked, how is it actually, what is its function in vivo in cells? And we have combined a very rapid depletion of SPT6 with occupancy profiling using MNet-seq as well as TT-seq, which is our labeling, RNA labeling protocol that gives us RNA synthesis rates. And in the occupancy profiling with engaged polymerase, we can see that upon depletion of SPT6, you get an increase in polymerase occupancy in the first nucleosome. So this is consistent with the idea that you need to exchange NELF against these positive chromatin transcription factors before you go through the first nucleosome. Otherwise, the polymerase has problems to go through. And to investigate these aspects in much more detail, we also set up a biochemical assay that now allows us in a very defined system to investigate transcription elongation by RNA polymerase II through a nucleosome. And that assay we now use to look at the effect of several factors. And we found that SPT45, which is the DSIF factor in mammalian cells, SPT45 in this yeast system, as well as TF2S, are definitely required for the polymerase to go through a nucleosome. You see this upper band here for the full-length RNA product. But when you include either FACT or CHD1, you get a strong boost in passage of the polymerase through the nucleosome. And that is even a little bit higher when you add both factors, but not much. But what clearly is the case that the CHD1 chromatin remodeler, it's an ATPase, as well as the FACT histone chaperone are strongly boosting polymerase transcription through the nucleosome in this minimalist system. So now we have a very well-defined biochemistry, and we can solve those structures by cryo-EM. These are all yeast proteins. And we have recently achieved that. This is not published yet, but you can read about it in bioRxiv. It was just put online. So on the left, we have a structure of the polymerase in silver with DSIF in green and the downstream nucleosome, as well as the CHD1 ATPase. And then on the right, we have a similar complex, but now CHD1 is replaced by the FACT histone chaperone. And you can see that the two factors bind to sort of equivalent or overlapping positions. So you can either have CHD1 or FACT, but not both, at least not in our scenario. I should say that these complexes were made by polymerases that would transcribe into the nucleosome, partially unraveling the DNA, and then the factors would be recruited. So these are real intermediates of nucleosome transcription, where the polymerase has begun to transcribe the nucleosome. And that makes it very interesting, because whenever you have intermediates, you can talk about the actual mechanism of the process. Now, what do we learn? Just a short summary of key points. For the CHD1, what we see is that the orientation, how it binds to the downstream nucleosome, is exactly what you would need to pump DNA into the active center cleft of the polymerase. In other words, CHD1 translocase activity would help the polymerase to overcome pauses that it definitely will have once it starts to go through the nucleosome. What we also see is that this regulatory domain here in red is displaced and mobile. And that is very exciting, because that led us to a model that polymerase transcription, or at least the entry of polymerase into the nucleosome, would displace this inhibitory domain of CHD1 and thereby activate the remodeler. And how do we know? We know from the work of the Bowman lab, which actually showed that this red domain is inhibitory to the remodeling process. And we know from our prior structure of the CHD1 bound to a nucleosome, which actually looks different. So it is the poised structure, but not the active conformation that we now see for the whole complex. So the poised conformation is formed by releasing this double chromodomain, which engages now with nucleosomal DNA. And that allows you to close the ATPase motor. The two lobes of the motor are now brought together in proximity, so they can actually bind ATP at their interface. So this is a poised conformation. It's ready to go. But the inhibitory DNA binding domain that you see up here in red will prevent the remodeler from moving on. At least it will not go full speed. And our proposal is that once the polymerase enters, it displaces this DNA binding domain, and then the remodeler is highly active in just that moment when the polymerase enters the nucleosome and not before. Now, in the second structure for the FACT complex, we see basically interactions that are quite similar to what Caroline Luger's lab just recently saw in their structure of a partial nucleosome bound to FACT. So we could use that structure. It helped us a lot to get to this complex structure, which is around 4 angstrom or so, whereas the CHD1 structure is at higher resolution. And so for the FACT, what is interesting is that we see what has been proposed before by Caroline, that the polymerase entry into the nucleosome and the partial unraveling of the DNA is actually required to then recruit FACT. So it will occupy binding sites here on the histones that only become available when you unravel the DNA, at least partially. So the idea is that then FACT can at some time be displaced by the polymerase when the polymerase progresses, and that will then allow FACT to, that's our proposal, to together with the histones be transferred to the wake of the polymerase, so that histones can actually be transferred from the downstream side to the upstream side of the polymerase, and thereby you prevent loss of histones and you prevent loss of the information that is contained in the modifications of the histone tails in active genes. So far, so good. This is what I wanted to tell you about our latest work on chromatin transcription, how the polymerase would go through nucleosomes. And of course, we're in the middle of this and need to do more work in order to get ideally a movie of the polymerase going through the nucleosome and how the factors help the polymerase to go through. But now something that is important subsequently, and that is, very new work from the lab, very exciting, which I want to share in the last few minutes. And that is actually addressing the question of co-transcriptional splicing. You all know that the polymerase will coordinate the pre-messenger RNA synthesis with the processing of the pre-messenger RNA. And that is, of course, capping, splicing, and the 3' cleavage and polyadenylation. And co-transcriptional splicing is the most elaborate of those three. It involves the large spliceosome, over 100 proteins, very complicated process. And when we think about co-transcriptional splicing, we basically think in that way. I think at least that's what you read in many reviews, that it's co-transcriptional because it happens on the nascent pre-mRNA that is still attached to the polymerase. But there's also accumulating evidence from several laboratories that it's more than this. It's actually a real coupling, a physical and functional coupling that somehow underlies co-transcriptional splicing, and that there's a feedback from polymerase to the spliceosome and back. But actually, the evidence is still very indirect, and there's no mechanistic information on how such coupling may occur. But most of splicing is certainly co-transcriptional, and the question is, if it's functionally and physically coupled, how exactly does that work? Now, don't look through that slide. It just tells you how complicated splicing is. But what is important for us today is only to remember that the first step is the recruitment of the U1 snRNP. You see here the green arrow. So U1 snRNP is recruited to the 5' splice site of nascent RNA. And that is the first step. Now, our idea was, can we probably try to get insights into the Pol2 U1 snRNP complex? And we joined forces with Reinhard Lührmann's lab next door, and they provided the U1 snRNP to us. And then Suyang Sang, who is really a fantastic postdoc, was able to reconstitute a complex, a transcribing Pol2 complex with U1 snRNP bound to it. And she could actually solve the structure. And the structure is extremely exciting because it's very different from what we see in reviews, because we actually see a direct interaction between the polymerase and the U1 SNRP. And this is just showing you that the resolution is variable. So it is a transient interaction. On the outside, we see quite a lot of flexibility, but actually in the interface between the spliceosomal U1 and the polymerase, it's highly defined. And this subunit here in violet, U1 U170K is its name, is highly defined, and it docks to RPB2, to the protrusion domain of the polymerase here in gold, as well as RPB12, which is a small subunit. So there's a direct protein-protein interaction between those two particles. And the pre-messenger RNA is tethering the two complexes, but it's actually flexible in between its two anchor points. So as the pre-mRNA comes out of the active site of polymerase, it will first form a flexible loop, and then it will base pair with U1 as an RNA. But the major contact is really a protein-protein contact, which is very unexpected and very exciting. So what does that mean for our understanding of co-transcriptional splicing? Actually, it led us to propose a model that we call the growing intron loop model. And that is an interesting model because it explains a very old problem in splicing, and that is, how can you bring together the 5' and 3' splice sites, which can be like 10,000 or so nucleotides away from each other, because introns are incredibly long, at least in mammalian cells. And the answer could be that by recruiting the U1 particle and the 5' splice site to the polymerase, and by retaining the 5' splice site near the RNA exit site of the polymerase, you could form a growing loop of nascent RNA as the polymerase moves on. And you could scan for the 3' splice site and the branch point, and basically match and bring those two splice sites together to lead to a functional pairing of the 5' and 3' splice sites on the surface of the polymerase. So our proposal is, from superposition of spliceosomal structures, that possibly this pre-spliceosome, the pre-spliceosomal A complex, forms on the surface of polymerase. That is the hypothesis, and that the pairing of the two splice sites occurs on the surface of polymerase, but that subsequent formation of a catalytically active spliceosome leads to the dissociation, and then polymerase II can go on, and the whole co-transcriptional splicing cycle would start on the next downstream intron once again, so you can start the next cycle, and splicing then occurs in the wake of polymerase on the nascent RNA. Now, last point, you can actually inhibit the process at A complex formation, and this is very exciting when we did that using this PLA-B compound, because what we saw is that there's also feedback backwards from the spliceosome assembly pathway to transcription. And just very briefly, what we see is that when you add this compound, then the RNA synthesis rates globally will go down. So not just splicing is inhibited, as was known for a long time, but also there's a strong feedback on active transcription. We see that with our TT-seq protocol in human cells. And when we look by MNET-seq where the polymerase accumulates, there's this drastic change that polymerase is no longer in the gene body, but it actually accumulates right in the beginning. You get a very large peak for promoter proximally paused polymerase. And actually the pause duration, we know this from multi-omics and kinetic modeling, the pause duration is strongly increased upon PLA-B treatment. So the polymerase now sits in the beginning of the gene, and obviously it does not get the signals that it needs to be converted into an active complex and to processively elongate. So that seems to be a very exciting mechanism, a feedback control. You know, if splicing is stuck, then pause duration is increased and polymerases are no longer released from the promoter. And we could even see that the PTEF-B complex through ChIP-seq of its cyclin subunit, that it's not recruited properly anymore. So you see here in the middle panel that there's far less occupancy with cyclin T1 upon treatment with PLA-B. So that's the take-home message for this new and exciting result. There's a very intimate coupling between pre-mRNA synthesis and the splicing of these pre-messenger RNAs. And a lot remains to be discovered on the mechanistic level. This is just the very first step that we saw that U1 and POL2 interact directly. And finally, I would like to thank the people who were involved in these most recent findings. Of course, in the first row, you see people who did structural biology. Carrie did start to move, our structural biology from the yeast to the mammalian system, and she got the polymerase diacif complex. And then Seychelles took over, and she could get the pause and the release complexes and establish the biochemistry for this step. She was helped by Lucas with the NELF pause complex, and Lucas is also responsible for the beautiful work of polymerase nucleosome complexes, also the most recent ones. He was now going to leave to Harvard as a professor. And so these three people really were at the core of establishing the pausing step as well as the release and transcription through the nucleosome. And then Su Yang is the fantastic postdoc who did the U1 polymerase structure. And Dimitri is the one who developed a lot of very good software that many of you probably also use, like the Warp package for pre-processing of cryo-EM data. And recently he also published a new package called M for multi-particle refinement, which boosts the resolution in many cases. Christian is essential for all our cryo-EM. He trains people, but he also is a very good electron microscopist, keeping everything running and helping people with their individual problems. And then for the functional genomics, Saskia and Björn figured out this switch, the CDK9 dependent switch, and the pause initiation limit at the beginning of genes. And then Livia and Sarah are responsible for the latest work, functional genomics of cotranscriptional splicing. And they were helped also by Michael to a great extent, a very experienced bioinformatician. So I would like to thank you all for your attention, and I'm of course happy to take any questions. Thank you very much.